It's time. We'll start, and if other people come in, that would be fantastic. But I'm really delighted and happy to see some people who are both familiar and unfamiliar. So welcome to the Precision Talks Scientifique Café Scientifique. My name is John Colburn, and I have the honor and privilege of coordinating the Precision Talks project. Today, the topic that we have for discussion later on is to talk about the legacy of the three projects that form ASPIS. I'll introduce that in just a little while, but as all EU projects that are both data producing as well as with the ambition of being transformative in terms of the use of that data to solve significant problems. In this case, the problem of, or the challenge of replacing animals in the testing of the safety of chemicals. Much of that legacy really resides in the data itself and how this data can actually be made available to all stakeholders. So that means that they need to be findable and reusable. And so the topic for today is to have a presentation from the data managers of all three projects to learn about the solutions that they have actually adopted for this particular challenge of sustainability. And importantly, to lead into discussion now and further discussions leading up to the ASPIS Open Symposium in-person meeting that will be happening in Maastricht between the 2nd and 3rd of July. Everybody who's attending this meeting is welcome to also attend this open symposium. Monica can send the registration information later. But we're coming to the end of these projects, and I think that's really important to begin to have these discussions on how EU's investments are going to be sustained in such a way. So I'll present first, and then I'm going to pass on the presentations to others, but I do want to give a brief introduction of what we mean by the ASPIS data and sustainability. So I'm going to share my screen. So the topic is data sustainability across ASPIS. And as I said, we're going to have presentations by the data managers of these three projects. The ASPIS cluster formed right after the three projects, PrecisionTox, OnTox, and Risk Hunter, were supported and were awarded by the European Commission. It's a large project, altogether 60 million euros, and each of these projects are quite distinct in their approaches to making that transition away from animal testing to a more mechanistic understanding of toxicology and the use of that understanding for the regulation of harmful chemicals. In PrecisionTox, it's all about leveraging evolutionary diversity to reveal the molecular basis of toxicity. And this is very much tied to comparative toxicology across different species, and I'll explain a little bit more about that. OnTox is a project that's more synthesizing existing toxicological knowledge to support risk, next generation risk assessment, very much heavily using computational approaches such as artificial intelligence and machine learning. And Risk Hunter is primarily focused on human-centric chemical safety assessment utilizing systems toxicology. All of these use systems toxicology in their approach to documenting the biomolecular basis of toxicity, but you can see that as opposed to PrecisionTox being focused on comparative evaluation across species, OnTox is very much focused more in terms of organ toxicity, and Risk Hunter very much driving force behind the use of in vitro model systems. For precision toxicology, the data that's being produced is quite diverse. And I'm just using that as an example because it's the one that I know best. But all three projects are quite both data rich and data diverse. In PrecisionTox, what we're doing is that we're exposing 200 substances across 5 model species, including the human cell line. And this involves C. elegans, the nematode worm, Drosophila melanogaster, both sexes, Daphnia, which is a water flea for ecotoxicology, and the embryos of Danio, the zebrafish, and Xenopus, the frog. And by doing these very well-controlled experiments, we are basically documenting what chemicals have what kind of adverse outcome in terms of phenotypes across these different species, but also extracting samples from these exposures from which we can have multi-omics readouts. And what we're talking about here is to be able to document the changing patterns in gene expression across all these species, making use of evolutionary knowledge of which genes are shared across the different species by orthology, meaning common descent, as well as comparative metabolomics, which is using mass spectrometry in order to measure the changes in the abundance of key metabolites. And so we have all this data that's basically having been generated, and therefore necessitating a data management plan that could sustain the investments that are being made in this project. And this includes, of course, the data that's associated with the chemicals that are being used for all these exposures. So here, PrecisionTox has created a chemicals library, and with it, a database. This library is made up of very well-chosen chemicals. I know that John Friedman is here on the call, who is leading this effort, and ended up with 54 pesticides, 87 pharmaceuticals, and a diversity of other substances, including industrial chemicals. But you can see that the majority of them here are in fact pesticides and pharmaceuticals, because of the fact that these are the types of substances that we have the most knowledge of their mechanisms of action. And via this database, users can not only basically explore the chemicals, but also they explore the chemicals in light of what we've learned from the exposures that have been carried out by these experiments. And these experiments represent a variety of different phenotypes. The first thing that needed to be done, of course, is to conduct some very stringent SOPs by which these organisms could be exposed to, as best as we can make it, similar toxicological conditions by which, therefore, the omics data would be comparable. And just looking at, for instance, creating these dose response curves across 200 substances, one recognizes right away that these different species have different sensitivities across the different substances. And this is all very well documented in a database unto itself, from which specific endpoints, and in this case, this is the concentration by which adversity is observed at a 10% level. And you can see that caffeine, for instance, you have a distribution of these benchmark dose 10 values, and in some cases being able to actually see some biology emerging from it. On the right hand side, you can see that biology is mostly reflected by the habitat by which these organisms live, or the exposure route that these organisms are experience through these exposures. And all kinds of data as well as comparable, that includes discoveries that are made on the relative sensitivities of the different sexes. Here's just a small subset of the chemicals that are demonstrated here in terms of the relative sensitivities of Drosophila male versus Drosophila female. And right away, you can see that roughly the majority of the substances, 72% of these chemicals, are more sensitive in the males than they are in the females. And the differences in those sensitivities can be quite dramatic, being different by orders of magnitude. And so it's very interesting sets of discoveries that really challenges our ability and our understanding of being able to do cross-species extrapolation. But there are far more comparative toxicology data that was produced by the consortium. For each of these species, different assays were conducted in order to be able to get some indication of different endpoints with human relevance. Danio, the zebrafish, the embryo, is the one that has created the most diverse set of endpoints. This is done by Stefan Schultz and the group at UFZ, where there are well over 50 different types of measurements that are being taken, whereas others like Drosophila or Daphnia, a smaller number of them, but very much tied to things like behavior, for instance, to be able to detect neurotoxicity and reproductive toxicity. So all this is a lot of data and must be basically not just collected, but also managed and archived appropriately. There are databases that have been produced by our colleagues, primarily at the University of Heidelberg. And here is, for example, the PrecisionTox phenotype dashboard, which demonstrates that across all those toxicological endpoints, we've accumulated 1.8 million phenotypic records from just under 130,000 different exposure conditions. And so a very rich data set, unique as being the largest harmonized comparative toxicity data set in terms of measuring the effects of substances across species that last shared a common ancestor almost well over 500 million years ago. And so it's truly representative of how toxicity affects animal biology. But that's not all, because in the course of the project, new workflows needed to be created. And here's just one example of a workflow that was created by a Phenome Center Birmingham, where it's not just the ability to extract the metabolites and the RNA from the same biological samples, but also how to make sure that the readouts that are coming from these samples are all harmonized as well, where the process by which the data was collected is all the same, and minimizing the amount of technical variation that could be introduced otherwise. And so these are very complex data processing and data collection workflows, and these must also be well documented and tied directly to the data so that it is truly fair, so that it could not just be reused, but also replicated as well. And so there are, throughout now 5 years worth of carrying out this project, a large number of new standard operations, all tied to the vast amounts of data that was collected. And this is a really good exercise for the consortium to do, because if these are the kind of steps that we had to take in order to produce the data, it's really important that these be shared at the end of the project as well. And so, for instance, we have a guide here to the PrecisionTox chemical library, and this also would, at the end of the project, necessarily be able to demonstrate the grouping and classification of the chemicals based on the PrecisionTox results, and link the substances to the phenotypes and link the substances to the putative molecular key events that emerge from the omics data. But also there's a need for the guide to the PrecisionTox model species, an SOP for all of their maintenance, a guide to the PrecisionTox chemical testing, so SOPs for the exposure, SOPs for the phenotyping, SOPs for the calculations, a guide to the omics measurements, SOPs for the sampling, the extraction, and the data production, and finally a guide to the PrecisionTox data repositories, the phenotypes, the transcriptomes, the metabolomes, and ultimately that integrated multi-omics result being represented as the interactomes. These are really important products, so much so that I'm demonstrating here to the left-hand side a guidance document that was published in September of last year, called the Guidance on Good Practices and Standardization of Sample Collection for Omics Analysis, an OECD guidance document that is members of PrecisionTox, including Professor Mark Viant and myself, have authored with others. And so if we're going to basically not just produce omics data, but make that omics data relevant for regulatory applications, all three projects have much to contribute from the experience that we've actually had in producing the data. That must also be sustained in a way that continues to drive the science and the application of that science forward. And so from this omics data, we've reached about the halfway point in terms of the collection of the transcriptomics and metabolomics. And here's just some data that's taken from the dashboard of the data commons. And you can see here, for instance, that out of 105 chemicals that we have the data across all organisms for both the transcriptomics and metabolomics, we're looking at 26,283,000 observations, and on the metabolomics, just almost 30 million observations. Those numbers are going to be doubling within the next 3 months. So a huge amount of data, and I'm going to keep saying this, a huge amount of data. And I think that we should all be proud of the fact that how we've managed through this funding to produce it. But again, the majority of the discoveries that are going to be made, are going to be well beyond the funding period of PrecisionTox in this case, and therefore the discussion that we're having today is totally important. And with that, of course, the useful tools for the data exploration. So here's just a snapshot of the PrecisionTox Data Explorer, where the metabolites here that have significantly changed because of the exposure to acrylamide are being compared again across different model species. And there's some really interesting findings by digging into the results in this way. Oopsie, sorry. So as I said, we're going to have this opportunity now to hear from the data managers of these three projects. And we'll start with Daniel Jenin, who will talk about what the solutions that he has devised with his teams for OnTox. And as I said, very different types of outputs, very different types of legacy pieces. Philippe Rocasera is going to be talking about the solutions that have been devised for PrecisionTox, and finally Barry Hardy for Risk Hunter. And with this slide, all I wanted to demonstrate is that in some cases there are some overlaps in terms of the types of data and the utility and of the data for discoveries. Other times they're very distinct but complementary. And hopefully by the time that we end up all meeting in Maastricht at the ASPIS Open Symposium, we'll actually have made a really good job at the integration of that across the three projects. And that's all from my presentation. And now could I ask Daniel to share what he has prepared for us?
Give me a second and I will share my slides. You can all hear me correctly?
We can hear you, yes.
Perfect. And now you can see it in presentation mode almost.
Good, we can see it presented.
Thanks for the opportunity to give a little talk here about the data sustainability in OnTox. I'm Daniel Jenin. I'm from Maastricht University. I'm associate professor there, and I'm the work package leader on the data management and quality control in the respective project. Now, I didn't put in a slide about the project itself, assuming that everybody knows what OnTox is about, but I will try, if there are issues with that, to explain where needed. So the data sustainability, but I prefer to speak about research output instead of data management, because it's research output that we create, which is more than only the data, but also the tools and so on, and so, as John already also nicely showed in his presentation. And that's part, of course, of the whole project management. So, and that happens not only during a project, but it already starts before you actually submit the proposal, and also afterwards, hence the sustainability. I will take you in a few slides back to my first start in toxicogenomics, basically, and we'll do that with a timeline, which is this one. And what you see here is basically, I started in 2007, and what you see here are the projects that I've been involved in, affiliated with, and the different time points that actually things like templates for toxicogenomics were developed and used. Of course, it all started already in the past with the Geo and Area Express databases, where in 2001 the Miami standard, so the minimum information for microarrays, was important. But for the years, several other developments occurred. So the FDA hired ArrayTrack, which was a database for uploading your microarray data and also do data analysis. So that was my first thing that when I started the job, was to go to the FDA, have a look at how that tool worked, and to implement it in our own field. I will take a pointer to make things easier. So I was also involved in custom genomics, large project on developing a prediction model for carcinogenicity and gene toxicity. And their main step was develop in the Netherlands. The large toxicogenomics center was created, but that actually started already in 2004. But based on the templates that ArrayTrack used, templates and a database were created for this particular project. Later on, already starting to develop in carcinogenomics, which started in 2006. But after it ended, IsoTap was available, and Philip was one of the investigators or developers of that. Data, important project which brought a lot of different data, omics data together from previous toxicogenomics projects like the carcinogenomics one. And there further developments were taken on, also with the IsoTap. In 2012, the MiniSec, so the minimum information for sequencing data, came upon. And after that, several other projects followed, which used bias studies as such. And now we are here with the ASPIS, starting 2001, and the Dutch large consortium on VHP for safety. It says 2025, because this was a slide from last year, but I should now say 2026 already, and it moves on in the coming years. So that's just a little bit of a timeline, and what you can see in here, and so what does that teach us? That's the important question. And all these templates and formats and databases, what they had in common was that they had a study description, had protocols which were used for getting the data, the sample description, and in the end also the sample data, so the omics data. So basically, it's always the same information in similar fields or boxes in these templates, and also each time a new database. Well, that's not convenient because we're doing the same thing over and over again. So what we learned from that is that you need to learn from the past. What did you do there? What was good? Take that along. So use what is available and add what is needed. And that's basically the idea also when we were writing the OnTox project that I fed in is to look at that and move on from that. So for the project proposal, so what infrastructure would we use for our research output, basically? So the central data repository was BioStudies, and the OnTox Hub, which was based on Cosmos NG, a former project, was to make tools and workflows accessible. So this was, before we started the project, the idea behind it. During the project, things changed rapidly with the changing AI, but still this was until the end of the project still valid. So protocols that we implemented, so the in vitro methods, we followed the OECD guiding documents of GIVIMP, GCP guiding guidelines, and for the in silico toxicology protocols, the QMRF and QPRF So that's QSAR prediction and referencing formats, these protocols. So what do we adhere to in the project? Well, open research data, so make everything public basically, uphold the FAIR principles, but we also put as text in the proposal is that after 5 years after completion of OnTalks, we will make the depository, the simple data repository, publicly available. So when we look at the consortium with an active research output management, one of the things that my colleague and I did was basically figure out which work package delivers what, where does it go, and what are the different depositories needed. Now, this is a snapshot from a few years back. Small changes happened, but not drastically to update it. You can here already see the bias studies that I mentioned, the OnTalks Hub, but also GitHub is mentioned here in AOP Wiki. So we have different work packages. One work package that looked at the biology, that would be work package 1, creating physiological maps. One on the toxicology side, so that was more on this one, this part. Chemical properties and kinetics indicated here, and then the 3 big work packages up here dealt with the case studies on the liver, the kidney, and the developing brain. So basically, these provided the names that would be fed in the different AI-assisted tools and workflows. This was part of work package 5, one of the other big ones, and the OnTalks Hub was more the outreach. Work package 13. So this covers basically almost all the work packages. I left out 2, which was the project management and the one that actually is like this Café, Scientifique, led by AltaTalks, so doing similar things in there, so more the outreach in that sense. Okay, for the central data repository I mentioned, bias studies, it basically is a place where you can put all your biological findings. I've put in the link to the paper, and also nowadays Area Express, which was used as only for storage for omics data, is fully incorporated in bias studies. So I mentioned that past projects already are in there. So that's the DIXA, the Hackathon, transfuse T, and Eurotox risk, where I have only the 2 mentioned from DIXA that I was involved in, so the custom genomics and the NTC one. So all these data are captured there, and that's also present in bias studies. So currently these have been added, so that's still ongoing, but these are the 3 that are currently also putting their data in bias studies. So a grand enrichment of data is present in this data source. Bias studies is from EBI EMBL. And you can keep the data private or make them public. As said, after 5 years of the end of OnTalks, everything will be public, but depending on publications and so on, you can keep data private. Some data are already public and some are still private at the moment. And this is the overview of what we have in there. We have different types of data in there, ranging from transcriptomics, tox stamps, but also NAMS data, so output data from 2 of the 6 endpoints basically that we were investigating. And this number is still growing, and 10 are already publicly available. Other repositories that you can think of, GitHub I mentioned, basically we have different layers. One is sharing basically the code and tools, so more for research purpose. We also have information and material for educational purposes, and the Other one, the last one that I've mentioned here, is the ONTOX physiological maps and ontologies. And speaking about the physiological maps, that also has its own website and its own connectivity, also through Minerva and Luxembourg. And this is become a large part actually, because it was actually intended as filling the gaps on the biology, but it turned out as one of the important milestones from our project. And the physiological maps basically is an overview of pathways, the connections between the different genes, proteins, and how they interact, and the layers that you can put on over them, and use this information basically to identify, for instance, key events that are worthwhile to put in your maps. Other repositories, and that is a list, of course, YouTube, where we have videos from conferences, webinars, tutorials, and so on. Zenodo, which can contain miscellaneous documents, which you can't place in any of the other databases. The AOP Wiki, where the ONTOX AOPs eventually should land. And the last are Teams and SharePoint for our project's documentation. The ONTOX Hub, I mentioned that, which brings a lot of the information together. So we have different resources, the physiological maps, the AOPs, ontologies, tools that feed in there, and also the curated data collections, which are in, for instance, bio studies, which are not copied over to ONTOX Hub, but will have a link and feed in there. And in the ONTOX Hub, there will be the tools and workflows leading to basically the services and expertises that are needed to move forward. Now, the plan is to launch this in the second part of 2026, so I can't say much more about it. Probably there will be a bit more information during the ASPIS Open Symposium on this, so worthwhile to definitely attend. Well, with these repositories, also templates and other useful formats are needed. For the bio studies, we created a tutorial and a template to make it easy for the researchers to fill in these templates and upload their data. The AOP Wiki, there we have also created a training document and a template, which was quite difficult because you basically need to be building an AOP live in the AOP Wiki, and then the information was extracted from that. So there's no real guidance or good guidance when you go to the AOP Wiki. The tox stamps, based on the Krebs paper, so they're extracted from there. There's a template with instructions, and we have also a clean template without the instructions. And in our collaboration with VHP for Safety, and large language model was developed, which could aid to fill in this template automatically, which is called the tox stamp assistant. The link to the paper is indicated, and this tool has also been launched also last week in the VHP for Safety platform kickoff, which took place last week Thursday. Then when integrating tools and data, of course, the ONTOX Hub is part of that, but that led to OPRA, which is the ONTOX AI-supported probabilistic risk assessment approach. This is still also work in progress and will, of course, be integrated into the ONTOX Hub, which has different beginning and different checkpoints and different information that will lead to whether you can consider a compound safe, or maybe it has some concern or high concern for their risk of using it. So after, basically all of the information that I've mentioned from the during will also go to the after part, but I didn't want to repeat everything again. The difficulty will be with the Teams and SharePoint, because that's something that is being paid by one of the collaborators. So the transition needs to be made to a more sustainable one. Same with the website of the ONTOX, and probably have seen that with many of European project or other project that in the end websites will be gone because you can't keep on paying for websites if they are hardly being visited, of course. Having said that, most of the databases or repositories that are being used are in a way sustainable because they will be on the long term still present. You never know what happens in the future, but still for now, they're good to go. When looking at the OnTox impact, which is already on our website, we have AI tools and software that have been developed and used during the project and will remain active. And this is the list that is present there, ranging from systematic review, making that automated using AI for that, using a large language modeling. The Tox Index, that's a platform for regulatory toxicology. Tox Transformer, which brings multi properties together in a prediction model. DocTox, which targets MIEs and organ toxicity from molecular docking. Then the ECSDT is a computational workflow to classify the small molecules following the ECCS. Then two on QSARs. The Vega QSAR is one that has already been developed, but has been now updated by new machine learning models. And the QSAR PsyChem is one for physico-chemical property predictions. That's a brand new one that's together with people from the VHP for safety on the disease maps development or the physiological maps. And this is then a large language model based system to explore these maps. And the last one is the Neurotox KPTGT, which is an AI pipeline that uses the Campbell activity by activity data to basically predict molecular initiating events. As I mentioned, the physiological maps became more than just an asset to bridge the biology to the AOPs and QAOPs. These are a few snapshots of the liver lipid model physiological map, where you have on the left side a grand overview of the liver and the different processes that happen in the liver, which can be broken down into smaller pieces, as indicated here, with all the different compartments. And these are actually displayed underneath this layer. And here you have, and it's really zoomed out, these are a network of so basically the pathways, as you may know it, network of the genes and proteins that are interacting with each other. And everything that is blue is an overlay of gene expression data for just normal liver. We also have the collaborators that's used impact, of course, the JRC, for which we have a collaboration agreement, and we have a memorandum of understanding with ASPIS, PHP for safety, and the University of Seoul. So collaboration with people from these has been great and is also ongoing. I mentioned PHP for safety already with the Tox Standard Assistant, ASPIS, Helena Kadorova, who is also involved in the OnTox project, is in the lead. So with that, we have nice contributions. Of course, what I didn't put on this one, huge collaboration is, of course, within ASPIS, but that I think is quite clear. And that was it from my side. So I will stop sharing and have a look who's still online and not sleeping.
Thank you so very much, Daniela. It was really actually very informative and very exciting to see your approach, but also your results. Wow, nice set of tools and resources that you're creating and leaving behind with the project. We're going to hold off on the questions until at the end of the speakers' presentations, and so I'd like to now ask Philippe to give us his presentation on his approach to sustainability and here for PrecisionTox.
Thank you, John. Can you all hear me okay?
We can indeed.
Yes, very good. So good afternoon, everyone. I'm Philippe Rocasera. I'm based at the University of Oxford, where I'm an associate member of faculty. And I've been part of PrecisionTox since inception. And I'm very thankful to John and Daniel for setting the scene so nicely for the presentation. So I will go over quickly something that has been already covered by John, but just to reassert and reinstate some of the key steps of how the data has been generated by the PrecisionTox consortium. So John told you about the kind of species that we've been working on, and also Daniel has covered an important aspect, which is the existing tox exposure data available and produced by Legacy Consortium. And I wanted to single out a couple of those. The Inomed PredTox consortium dataset that had been formatted to the ISA syntax, and as well as the EU carcinogenomics project a few years back, which was at the which funded the initial work on the ISA tools and the ISA component. So this is an important aspect, as you will see later. So when it comes to PrecisionTox, yes, we've been testing about 200 carefully selected chemicals, which is a fairly large collection of representative molecules, scanning an array of chemical classes for which we have a lot of information. And these chemicals have been tested using different data modalities. So we have three molecular dimension modalities, two on the metabolite side of things, using mass spectrometry, two different acquisition, so helic and lipid modalities, as well as the RNA-seq data profiling for these to understand the transcriptomic response following exposure. But also, as it was pointed out earlier on, a significant effort has been placed on routing also the biological response into phenotypic observation with a particular expanded register of phenotypic response in zebrafish. The work itself has been split into two major streams in a way, or actions. The first one was a pilot study, followed by a production study. Unsurprisingly, the pilot study led us to the establishment of all the dose response threshold, but also allowed the group, the consortium, to establish all the procedure, validate really the SOPs, understand the error models as well to an extent, so that all the learnings that could be passed on to the production study, which relied on a slightly different design, but informed by all the information collected during these initial phases. This has an impact because from the start, the PrecisionTox project wanted to implement a FAIR by design approach. This is a kind of a play on word on two aspects. We wanted to make sure that the notion of FAIR, the FAIR principles, were from the inception at the core of all the data management practice. So from very early stages, we wanted to make sure that the data would be self-describing, so that we have an ambiguous and exhaustive descriptors available in the long term for the data. We wanted to make sure that we had explicit semantics. It's just not strings that we are annotating the metadata with, all the data, the findings with, but we can also make the most of the huge knowledge that's been captured into ontologies and vocabularies that are used to really build these knowledge graphs that are really at the tip of the scientific work, especially in the context of application of AI at the moment, which is essentially the fuel that is needed to correct some of the imprecision resulting from using LLMs, for instance. And so essentially, it may also mean being able to deliver machine actionable data sets. And by this, it means that agents can find the data sets and make sense of them without human intervention. And to achieve this means relying on the previous two steps. Another key aspect to the work by considering a fair by design approach is to establish trust in the data by ensuring that the provenance is embedded into, so you understand when you grab a data set which software tools have been used to transform the raw data and how the data has been acquired from samples collected in the lab, and you understand the audit and trail of the different samples collected from the different experiments. So this is a kind of ability to transition from the wet lab to the dry lab without breaking the path, explaining how the information is accrued over time. And this builds into the longevity and legacy of the data and the investment consented by the EU in the ASPIS projects such as PrecisionTox. So this is a kind of duty to ensure that the metadata record exists and is annotated to the highest standard. The other aspect which is interesting in the fair by design approach is that we tapped into the study design information that was established by the different working group within PrecisionTox. You remember the distinction that I made at the beginning between the pilot study and the production phase. We totally exploited that information. I will get back to this on this point, but here. In terms of having the ability to deliver a machine actionable data set, it means that we also try to make the most of what has been already produced and is available within the community. And this means performing a state of the art in a way, promoting reuse, identifying the different data formats and vocabularies available and pertinent to the field. And this is simply to prepare the gear up towards potentially the transformation and the rendering of all this information to RDF and RD knowledge graph, which represent the most mature state of fairness to an extent. So this is really at the core that has been driving the work in the consortium. As well as to deliver a data release plan and the cataloging capabilities to ensure that metadata records of each of the single data set produced by the consortium can be delivered to help the discoverability and reuse of the assets produced by the consortium and beyond the life of the consortium, which is an important point. So the pilot phases has allowed us to test our procedures, and because this is really the difficulty that all projects faces when starting and rolling. There is a kind of difficult period when the data is being produced, but yet the tools that are usually meant to be delivered by the different specialized working packages in a different consortium, these delivery usually take time. So there is a kind of misalignment between the need and what is actually taking place in real life. So this is the danger, and how do we mitigate those risks? In the context of PrecisionTox, we started relying on spreadsheet-based templates. This is extremely pragmatic, embedding naming conventions, which have been encoding of the data, so that we could deconvolute based on this naming convention, the exposure, the nature of the exposure, which compound was used, and we had a coding system. At the same time, in parallel, we had the ability to develop a metadata tracker system. It's just to collect the user requirement as well during that phase, which was extremely important. Talking to the different subject matter experts in the consortium, understand the need, and develop the software solution prototypes, as well as in the meantime, implementing all these different efforts in software engineering. At the time, that was before the revolution that we currently know in terms of coding, but implementing the best standard for the the code documentation, the code delivery, and the integration of the different tools. So this led to the development of a tool which we call the metadata manager tool. The file transfer, because obviously owing to the nature of the data, especially the omic data, we need to have to rely on to decouple the file transfer from the metadata tracking, and we chose the Globus file system. I will get back to that point later on. At the heart was also to understand that different users have different needs. So there is no one size that fits all. And it's important to be able to deliver a different format for different or distribution, as it is known in DCAT jargon, for the same information. So the project can kind of provide the serialization of the data either in tabular, tab format, JSON, but also we looked into providing a context file from the JSON, which provide initial capability for the RDF representation. So I will go quickly through the next few slides because this is simply to give you a feel of the user interface of the PrecisionTox Data Manager, which has been built by exploiting the information about a study design. So we users from the different data production center had simply to rely on to provide limited information, just to what the essential information that was needed. All the rest was already behind the scene coded. So we had the ability to reach annotation by stealth to an extent. We don't ask people to mark up the compounds with an accession number because we have done this pre-work before, and it's the same for the various metadata that was needed. So this is just quickly showing you a few screens, which generates a Google spreadsheet, which could then be shipped, exchanged between the different funders. But at least we had clarity, and you can see the first column here, the coding, the hash coding that was delivered from the different metadata element that uniquely identifies each of the sample that would be produced. And it was used as label in the lab, in the wet lab, for tracking the sample. Of course, this tool is backed up by an API, which means that we can directly interact programmatically with the endpoints, and we also have that information somehow decorated with more annotations and ontology terms, as I will point later. So in terms of developing software and why we did this is simply to address the gap that I was mentioning between the moment of data acquisition, data production, and data transfer. So we need to have the ability to structure the metadata early on, transfer that metadata in kind of ways that are workable in the lab. So this is the red section here. There is this template that is filled by the user, sent to the lab. In the lab, people are tracking this information and filling up the missing metadata that is only generated when you execute the plan in the lab. And then this in data sample collection step is realized. So you have the combination of all the metadata tracked in this document that ships along with the actual physical physical samples to the data production centers, which were Birmingham and MGI, and mostly Birmingham. I will spare you from the tribulation of the project here. But the key point, the receiving partner has this kind of full manifest of the data, of the metadata, with a complete understanding of the information required for the understanding. And this will be exploited later on for the deposition. What we explored in this approach, in the PrecisionTox as well, is to understand, because this is an important part of the FAIR principles, especially on the aspect of the interoperability, focusing on the community agreement regarding the metadata. So I'll give you simply one example here. On the alignment of the OECD omics reporting framework, or the guideline I think it's 390, which we use to anchor or augment the default EMBL metabolite configurations that I'm showing now on the screen. This is an effort maintained at EMBL EBI, so that we make sure that we can represent and marry the two requirements. The generic requirement from the generic repositories with the specific, especially I highlight here the QC aspects, which have been embedded into the PrecisionTox specific code as key information to provide, especially for the mass spectrometry applications. And this is essential to the long-term reuse and repurposing, providing this information, the raw read without the raw spectra without necessarily the QC elements limits to an extent the possibility of recorrecting. So we made sure that the PrecisionTox metadata and data contain that information at reposition time. This is just simply to highlight as well how we relied on the ISA API. So this is an existing library that had been developed by previous multi-omic tox exposure funded related projects, as Daniel mentioned. And here I'm highlighting the set of resources from Jupyter Notebooks to also documentation and APIs about the ISA API, that how you can use it to structure the metadata and build a metadata graph that links the data and all the annotation that you can use, as well as documenting how the annotation with ontology term can be done. And you can query the resulting metadata file using ISA GraphQL, for instance. So I don't have time to expand on this, but this has been a main driver. Still promoting the adoption of and the convergence of the practice. This is exploiting work that had been done within the EU as well, supported by the IMI projects, the FAIR Cookbook, which looked at how we can package metadata and data using our research object crates. And we do that programmatically. Again, so Arrow Crates is a resource that is gaining momentum that allows you to really package information very effectively. And it's a thin layer that you can augment with the metadata payload of your liking. And we have worked with a group to develop an ISA Arrow Crate profile. So this is really exploited to the full. We can reference workflows which have been deposited to Workflow Hub through a DOI and all these kind of things. Now, when it comes to the PrecisionTox code and data legacy, the code of PrecisionTox, most of it is available through PrecisionTox GitHub repositories and will be released on due time. The releases will be identified by DOIs through the integration with Inodo natively from GitHub. And whenever possible, the execution of a particular workflow is referenced from within the metadata, and the workflow is identified by its DOI, minted under Zenodo. Here we need to highlight that we have been working closely with Elixir to develop a specific component that manages the reposition to an extent of the PrecisionTox ISA coded data to EMBL repositories for the modalities that are supported by the EMBL repository, namely transcriptomics and metabolomic applications. The interesting bit on the infrastructure that we built here is to first rely on existing EMBL services. Don't want to build something that already exists. We just want to exploit that infrastructure, but we still need to have a thin layer of conversion to be able to dispatch the metadata and code it into ISA into the relevant repositories. At the same time, we want to make sure that we are able to write back the accession numbers minted by each of these individual repositories back to the original ISA metadata file, so that we can persist that information and without losing the fact that the ISA is the only archive that links all the modalities together. because of course the weakness of these major repositories is that they are technology centric. So some of the representation would be lost otherwise. And then in the context of the phenotypic data, because there is no such repository, specific repositories at EBI, then we need to rely on generic repositories, such as Zenodo. And again, we can do this programmatically, and we can push this information to the Zenodo, get a DOI, which is providing the longevity for identifying the resource, which ultimately is what matters. So John already showed rapidly the work that is being developed, and it will be made again more widely available on due time. All the credits goes to Gaurav Divan and Juan Carlos at Heidelberg. And I'm just a teaser here showing some of the representation that allow you to see some of the results, the incredible results and data sets that have been produced by the consortium, just a voice of this consortium today. And I wanted simply to highlight that under development is an API as well, which should serve some of the data as JSON payload, should you wish to exploit that programmatically. But of course, when it's, and this is another view of additional data, I don't have time to obviously delve into, and this would be the topic of a different project. Anyway, so going back to the notion of data of AI readiness, I think I wanted to simply highlight with this very simplistic cartoon, what would be the situation without taking into account the resources that are available in terms of vocabularies and resolvable identifiers. This would be the kind of structure that we could expect from with using simple textual strings when representing some of the output of a project such as PrecisionTox. So of course, if we look at the person, we can identify this is probably a person, but there are probably many Martin Jones on the planet, and there would be ambiguity. Then again, PTX 026, we don't necessarily know just with that string what the compound is, and so forth. So the main advantage of having done this mapping, this alignment to identifiers, first of all, we can replace or associate those strings with identifier which are HTTP resolvable and regularize, as you can see, using identifier.org, and we can understand the machine, and this is probably the most important point, the machine can understand that each node has a particular meaning. This is the semantic aspect of this integration or the realization of the FAIR principles in action. So each node that you can see here has a type: investigation, publication, organization, person, chemical entity, and this is really what is at stake here, to make this explicit to machines. And this is why we mention all this preliminary work that is necessary for structuring the information. So things, not just strings. You can resolve and request for more information for each of these nodes. So as a conclusion, I think in terms of, if we look at PrecisionTox data and services from an AI readiness point of view, we've started with a very detailed data management plan. That has been the kind of paramount key starting point, which has been implemented as a kind of FAIR by design approach throughout. We are attempting to deliver what good looks like when it comes to tox exposure data in terms of fairness. But at the same time, we are aware that not everyone is necessarily fancy digesting RDF or link data. So we are also providing the data sets in more classic, I would say, shape distribution of data. But the good is also available for computational agents, should it be. And last but not least, I think what we strive to do is to lean into the existing EU infrastructure, existing standards, existing vocabularies or syntax, because the cost of creating a new one is sustainability, and it's not guaranteed. So by reusing existing, we apply the FAIR principles again to those standards, and hopefully this will create a pool of data that is large enough so that we create accretion points and convergence. So I'll stop here, and these are all the people I'm indebted to, and I would like to acknowledge. And I'll stop here. Thank you.
Philippe, as usual, wonderful, wonderful, wonderful. Very exciting to see in its entirety. We're starting to get some questions now being posted on the chat. I encourage more people to do so, and I would like to now invite Barry to finish us off with his journey from data to knowledge to trust. Thanks, Barry, for doing this with us.
You just let me know you can hear me and see my screen.
We can indeed.
Okay, very good. All right, so I'll continue with what I would say is a reflective talk. I tried to provide links to further detail, and there sometimes I'm looking backwards to some history. Sometimes I'm looking forward, and it's speculative, and those are my own personal opinions. I will include, of course, some of the work we've been doing on Risk Hunter and our interactions on ASPIS and what its legacy can be. First of all, using this quote from Isaac Newton, we are always building on previous work. We're standing on the shoulders of giants. And I think from ASPIS going forward, we also have a lot of potential to build on all this wonderful data that has been generated on the project, but also what we've learned and how we go forward with that learning. And the point in this comment here is there is a limit, no matter how much work we put into our data management, our creation of fair knowledge resources, there is a limit to what we can do on that. A lot of knowledge remains very tacit and human. And I even think, despite the power of current AI, it's still rather limited in terms of real understanding, really interpreting evidence and making decisions. And we should keep that in mind as we increasingly include valuable knowledge from AI in our work. On Risk Hunter, there was a lot of NAMs development, both in vitro and in silico within Risk Hunter, particularly applied to human related endpoints. And there, early in the project, a significant initiative was this ASPA workflow approach, which was subsequently made into an ASPIS initiative, as we realized in the end, this needed a stronger community outreach and engagement, I think, an important legacy to take forward. And the basic idea was that we should structure everything we've done in our workflow responding to a particular problem formulation and be very clear what we have done, what evidence we used, whether it's in silico or in vitro, and how we interpreted it and how we got to a decision, and how that also is iterative and tiered. We built on previous work. So in Risk Hunter, we didn't start from scratch. We built on the knowledge infrastructure we'd been working on in the previous related project EU Tox Risk, where we developed this knowledge infrastructure that was reported in an article a couple of years ago. We have a current one under review, which updates this and some of the more recent work on Risk Hunter. But basically, we built on what we had before in terms of principles and implementation and added to that. And I think that's also going to be an important legacy aspect of building on all this very valuable knowledge infrastructure that we're presenting here today between us from the projects. There's a lot of work going into creating such a fair knowledge infrastructure. This standardization, this harmonization, the attention paid to detail on the metadata takes a lot of effort from everyone, but there really is a value release from all that work downstream when we use that in our risk assessment. When others in the future build on the work that we've created, and I think we have to acknowledge all the hard work done by many hundreds of scientists to build this kind of knowledge infrastructure. And the value is being released during the projects, but certainly can be released going forward. This ASPIS workflow approach was reported in this preprint in the last autumn. There's a paper now. The final paper is on Altex, and I think it's a very readable paper if you want to find out more about the thinking behind it. Yeah, I recommend if that's a new topic to you, it's an important one where we try to make the use of data in risk assessment well documented and structured so it's evidence-based. And when we have documented workflows like that responding to a particular problem formulation, which could be in human health, but can also certainly be in environmental health or safer by design, then we can look at how AI can assist us execute that workflow operationally. And the AI of today can certainly assist us in helping reduce the effort and time required to do certain tasks like do a knowledge mining of literature or complete a template and so forth. It can also bring evidence into the workflow for our consideration, but it assists us. It does not replace knowledge work by humans. It doesn't replace understanding and interpretation. So I'd like to just point out, even if AI agents are increasingly able to execute tasks, this understanding of what evidence we have and its limitations, its interpretation is a very important part of such workflows and the learning loops we have in them. Of course, they need to be executable as well. So such ASPA documented workflows also need to be computational. This is an example of one we built for hazard characterization. So such a workflow should be fair. So the fair data, but also fair knowledge. If we use data from models or from AI, it should also be fair and well documented, and we should have a memory of everything that we obtained in a particular execution of a workflow and decision. We also had a significant amount of innovation output. We've been running an innovation radar within the project. These are 4 innovations we submitted for review at TRL 6 to the EU and were invited to the Innovation Prize last year. So it's Safer World by Design, which is our platform for operationalizing and commercializing the knowledge infrastructure for NGRA, that's primarily NAMS-based itself, the ToxStem software for documenting in vitro methods, and the ASPIS software for documenting these NGRA workflows. But I should mention, in addition, there was a lot of additional related or supporting innovations. We had 26 in total, and that's an important legacy we are working on building on as well. This is the software. Again, we were building on the software from EU ToxRisk for documenting methods using according to the ToxStamp approach. That's in this Altex article from 2019. But within Risk Hunter, we've also now added a review process that's integrated into the software and the ability to comment and export reports. And this also is a facility that's available openly and publicly now to further projects downstream. So again, an important legacy that we can build upon going forward. As we work on other projects, for example, in the safe and sustainable by design area, we also see the limitations at times of the work on NAMS-based chemical risk assessment. For example, when we look at industrial products and advanced materials, polymers, graphene materials like we look at in this Accords project, there are also many additional aspects we have to address. So extending the knowledge infrastructure to materials characterization and doing an assessment on nanomaterials, for example, needs further work, but it's an example of how we can build related knowledge infrastructure that we want to be interoperable with our NAMS-based risk assessment infrastructure. So we have a couple of webinars planned on this topic for this knowledge infrastructure this month, and can also be part of the related legacy from ASPIS. Quite some time ago, the first European project I coordinated was started in 2008, and our first article on describing the principles for predictive toxicology was 2010. So I thought it was a good idea over the last couple of years to create an updated version of that. And I presented it at a couple of the Open Talks meetings, but we've now released a preprint on it, and we tried to build on the learning since in research related to new approach methods, and also a significant section on principles to consider in our increasing use of AI agents and orchestration on workflows. It's available as a preprint, and we're working on that in June to finalize an article. And I expect a series of related articles in times ahead on this. I think somebody doesn't have themselves muted, by the way, if they can mute. A recent tool was actually based on the learning we had within Risk Hunter, but also the ASPIS working group, where we worked on building knowledge graphs to try to support mechanistic interpretation. In a particular area in the ASPIS working group, for example, we worked on building a knowledge network for steatosis. And what we found is it's actually quite challenging and time consuming. And so in the end, we decided we'd create a tool that would have human interaction, but would make it more straightforward to build a network model based on current knowledge, for example, from the AOP wiki, to build a knowledge network across the mechanisms that you're investigating in a particular case study. And then to once you have that, to link it towards data analysis like you might have from a toxicogenomics workflow. And this tool, the second version, is available for exploration, and we also released a preprint on this spring. And it follows this workflow for creating the knowledge network from existing knowledge. And then, of course, you need additional workflows to look at your data analysis against existing knowledge. And where is the evidence congruent? Where is it contradicting? If we want to take an evidence-based approach, you need to always consider what we currently know and what does the new data say against that. And that can be quite difficult to do without such a knowledge framework. So this tool is available also for you to try if you're interested. At the end, we have to bring all this data and knowledge together for end users. This is our software that was initially designed with industry for risk assessment, and we've been more recently within SSBD projects extending it in our development version to also include evidence from safer and sustainable by design, sustainability, and circularity, so that the knowledge becomes even more heterogeneous, the decisions become even more complex. And there, again, the AI can assist us with clear business cases on the amount of time it can sometimes take to pull the information you want to use from documents or literature, or the effort needed to create reports or variations on reports. So the AI can help assist us on practical problems like that. In terms of using data and knowledge as evidence, I personally like the Bayesian approach because it allows you to weight evidence in the models you build. This is an example from a collaboration between ourselves and Bayersdorf, which is deployed and used in our risk assessment. But I do see such methods being used more as we need to support more multi-criteria decision-making and more complex decisions. So it's not just based on a personal opinion. This is work from OpenTox as a research project where we've tackled the problem of trying to build models across species, and I think could be very interesting to look at this valuable and extensive data from PrecisionTox to see how we can enhance such models. And this is now an ongoing initiative within OpenTox, and again, an example how we can build on the legacy within communities. Just pointing out, there's a lot of very interesting work currently happening. I'll note here the work from Ivo Dezhorsky on how he's using AI agent technology to carry out tasks like using data from models like the OECD toolbox and make that more tractable. But we really need to understand when we have such systems what actually is happening, and not just take the outputs, but understand everything that's happening in the workflow. So transparency, interaction loops on learning, again, very important topic. A new initiative I started in Risk Hunter last year is what I call the Open Model Repository Initiative. And again, I'm going to open this up probably through OpenTox as an open science initiative with the goal that we increasingly are using evidence from models. How do we document the protocols and templates for them? So we have some proposals on protocols, but from simpler models like QSARs through to more complex models like quantitative AOPs. And I think, again, that's a valuable legacy for us to build upon in terms of increasingly using models, including AI-assisted evidence in our workflows. One example of that is if we use a quantitative AOP model, it may involve experimental evidence, machine learning models, toxicokinetics models, and then the quantitative AOP model itself and its description. And that all needs to be described very carefully. So if we are using a result, like a point of departure for a particular mechanism and its interpretation, we have to have that well documented if we want to consider that as evidence that a regulator would consider in our submission to them. Message from this ESA Talk workshop based on this topic of fair data was, I would say, it strongly comes out across stakeholders that the use of fair data and knowledge is an absolute essential aspect of trust if we're going to build AIs based on background knowledge. It's an essential aspect that we need, and it needs to be very clear which data and knowledge is going into the training of the AI if we're going to trust it in our risk assessment work. Sort of a summary of these aspects that we bring data and knowledge together into workflows, with documenting always where they come from. Knowledge graphs enable us to connect relationships between knowledge is important too. And we have to bring those into assessment tools that end users can use, and also deal with conflicting evidence and information and their uncertainties. That should support our move from traditional toxicology into what we can expect maybe the future regulatory science framework, which will be based increasingly, we hope, on what's currently a voluntary approach, but it's embedding safe and sustainable by design, which includes NAMS-based risk assessment, but also other evidence like from life cycle assessment into an evidence package in which we can use in our current product innovation in industry, support all the SMEs that are developing innovative products, and eventually have this be a new kind of way of evaluating evidence when it comes to regulatory agencies. And that means embedding the use of such evidence into the product innovation lifecycle and decision-making made in early product innovation. I think the current designs of AI, even if they can do a lot, are limited. And I've called that this rather colorful name of an AI that loves, but I don't mean a romantic kind of love. I mean an AI that really understands this kind of integration with humans and the collaboration that we have in our pursuit of truth and wisdom, our service to the ongoing community, and our use of knowledge to make wise decisions in the end. And that requires, I think, the principles like we outline in the OpenTox 2.0 framework I mentioned earlier, but also additional principles such as human agency, governance, stewardship, and wisdom. We need ongoing investment in this knowledge infrastructure we've developed, both private and public, to release all the value from it. And I think there's a lot of opportunities to have a strong European infrastructure, including this integration of AI, especially a more intelligent, collaborative AI that can support our understanding of the evidence that we're generating. Governance, I just want to say, I think governance is really important, and I've been working on that in a couple of different ways on OpenTox and Safer World by Design, but I think there's a lot of governance issues we have to address as well in terms of this, not just data governance, but our approach to sustaining these knowledge resources and our interactions as a community. And the AI is not going to teach us to be wise and make good decisions. So we need to be humble in the knowledge towers that we built and realize there's a lot of additional aspects that are human-based to choose wisely what we do with this data. Finally, to finish up, knowledge is not the same thing as understanding, and understanding is not the same thing as wisdom. So our legacy will be taking this data knowledge forward so that others can build on it and enhance the garden we've created. Final comment from Magister Ludi by Hermann Hesse. He explores if in this novel of his the goal of academic research in generating knowledge, but he does remind us, as in this quote, that there are constraints and limitations on that, and we have to, in the end, accept contradictions and uncertainties as different poles of a unity, and that certainly is the case, I think, in our field. So with that, of course, acknowledging all the great collaboration within RiskHunter, OnTox, and PrecisionTox, our funding from the European Commission to do this work, and also continued in current SSBD projects. Thank you for your attention.